Buongiorno.
Good morning. Hello, Rita.
How are you, Gloria?
Very well, thank you. And you?
Good, thank you.
Just few minutes and we will start.
Okay, I saw the
Andate a casa, non c'è nulla da fare. 25 anni fa, una diagnosi di distrofia muscolare di Duchenne.
È vero che i miei muscoli diventano sempre più deboli.
Una patologia degenerativa rara che colpisce i muscoli. Ma c'è chi non si arrende e fonda Parent Project. Intanto il mondo cambia e anche Parent Project cambia un po' il mondo.
Ringrazio Parent Project per
nuovo approccio terapeutico
dall'ultimo anno finanziato da Parent Project.
il project che ha
circa 8 anni, è la prima volta che
in maniera
I pazienti oggi hanno un futuro da immaginare, degli obiettivi da raggiungere e dei traguardi da conquistare. Oggi, anche con la Duchenne puoi diventare ciò che vuoi. Come
Luca
Luca,
Buzzella, che ringrazio per essere con
Morire insieme, in fin dei conti, è una sfida già vinta.
I'm here to tell the future is there.
Jacopo,
alla Duchenne.
Matteo, Lorenzo, Luca, Samuele, Francesco e come tanti altri ragazzi che vivono una vita piena. Ma questo non basta, non c'è ancora una cura e tanti sono gli ostacoli da superare e le strade da aprire.
Tutti insieme abbiamo formato una grande famiglia.
Scrivi i prossimi 25 anni di storia insieme a noi.
Buon pomeriggio, bentornati.
Good afternoon. Good afternoon. Welcome back for those of you who were here yesterday, but welcome for those who have just come to meet us here. I would like to remind you all that we are in our headquarters of Parent Project Italy. Well, we've just organized this place as a TV studio, you know, so it now goes as it goes for all TV studios these times. You can see we're not wearing masks because we've all been tested both yesterday and today, therefore we can do without a mask. So we have some 500 people who have registered to our international conference. Of course, we used to meet, you know, in person, but it is as though we were in person, if you wish. Let me tell you how it's going to work. Well, first of all, for you to attend the live conferences, just go to the website conferenza.parentproject.it, and next to the title of each session, you can click Watch Live. And our conference, as you might know, has simultaneous interpretation. Therefore, if you wish to listen to either English or Italian, you can just click on the interpretation icon, right-hand side below with the Zoom screenshot. But please let me remind all the speakers that in order for our interpreters, whom of course we wish to warmly thank, and they thank the speakers, but in particular Vanna and Alessandra, because as we said yesterday, they have been with us since our very beginning. So anyway, for them to be able to work properly, thus allowing all listeners to follow the presentations, please remember to speak slowly. I would also like to say that all speakers should use a computer, not a smartphone, not a tablet, and please, your earphones should be connected to the computer. Please don't use any Wi-Fi or Bluetooth because otherwise the sound will not allow our interpreters to work properly. One more thing, again, a very important thing. Yesterday we were very good because we seem to keep the time schedule. Hopefully this will be the case today and in the next few days too. You can see Filippo Buccella here next to me. He needs no introduction, of course. Here he is. He is the founder of a parent project Italy, and he for 20 years was the president of the association, and he now holds a very important position, if you wish. I mean, he plays an important role within the international parent project and also deals with the scientific part of our association as well as its communication. Ilaria Zito is sitting next to him. She's one of the 3 biologists working for us, together with Gloria, whom you already met yesterday, and with Fernanda, who is behind the lines, if you wish, but you will get to know her tomorrow. She, together with the 2 of them, is responsible for all our scientific activities. Ilaria more specifically deals with the patient registry. So before giving them the floor and allowing you to attend the rest of the conference, let me just ask all the Parent Project Italy participants, but of course we would like to welcome those who are listening to us from around the world, to please fill in a questionnaire, the one that I'm sure you have already received. It is very important because it will allow us to evaluate the impact of a project that was closed down last December the 31st. Yesterday, we closed our daily session with some presentations about our fundraising activities, whereby we can actually sponsor all our scientific projects as well as at least partially our support activities for families. There are projects that we may embrace thanks to our participation in the different government applications. They are of paramount importance, and therefore we need you to fill in this questionnaire, please. There's also a QR code in case you want to use it to access the questionnaire, but the link will be placed into our chat, into the chat of this very meeting. So thank you again. Thanks a lot. We're very nervous, you know, today too. Anyway, Filippo Buccella, you have the floor. Thank you, Cristina. Thanks. Well, first of all, I wish to congratulate Cristina on being so professional. Actually, she is, you know, playing this new role for her very professionally. When she first came to work with us, she never thought she would once have to be involved in such activities, but apparently she's very good, and the same goes for the rest of us. Actually, the association, especially in this last year, has become a real TV channel. You know, we actually broadcast an update of our activities on a daily basis. This year has been particularly oriented to this, even though we have been doing that for several years now. But we've been there for the last 25 years, you know. We are almost celebrating our 25th anniversary, and actually, it goes hand in hand with my son Luca's 30th birthday. Well, both dates actually speak for the time that has gone by since the Duchenne first came into my life and into many of other people's lives. So our association has been growing thanks to the daily activities, even small daily activities that we've all been engaged in as families, as boys, as researchers, as the Duchenne community. This is how we like to call it. Of course, also thanks to our staff being behind our back for so many years. We've all learned to do things that we never thought we would once do. So that's maybe a destiny that we thought at first we would have to accept without having, without doing nothing. But now we know that there's something we can do. So it's good to think that we can change the world. And it seems like, at least within our framework, there is, room for improvement, mainly helping the families where DMD diagnosis arrive for the first time. And this has been the case for the last 25 years already. So actually the conference as such is getting 18 soon. And of course, shall we talk about the differences between its beginnings and today? Well, of course, we may even not do that, because at that time we knew nothing about so many therapies. We had many doubts about cortisone. We knew nothing about the role of physical therapy. What about prevention in the respiratory function? There were no guidelines. There were no patient registries, not anywhere in the world. I mean, so many steps forward have been made thanks to the families' cooperation. And again, it is paramount. It is incumbent. We should understand the relevance of such cooperation being relevant. We must really go hand by hand towards the final, the ultimate goal, because we have a difficult rival out there to be beaten. So this is our 18th conference, and we're here to talk about advanced therapies. Now, when I was told that I should be speaking as chairman at this specific session, advanced therapies, well, of course, I soon thought of cortisone or other drug therapies that may regard other diseases. So are they advanced because they are old? Is this what we mean? Obsolete? Ilaria, shouldn't we understand the idea of advanced therapies as obsolete therapies, as somewhat older therapies? Well, not quite. Actually, advanced therapies really account for the last frontier in scientific research. There's much turmoil, actually, because highlights are being, spread everywhere in this field. They are actually new generation therapies. This is how we should call them. They do not use a synthesized drug, but biological products. So they may be based on RNA, DNA, cells, or even tissues. That's what we mean by that. Something very innovative, especially compared to the time when you first came into this world, Filippo. So novel therapies with huge potentialities. So if we use apparently such biological elements, we can actually correct or even replace, if not altogether improve the dystrophin gene, which is responsible for DMD. Now, today, the claim of our conference, you may have realized that Filippo is moving towards one direction, even if from different walks of life, so to speak. Today, we are mainly going to go through two walks, but first, sorry, let me expand on the advanced therapies. What do we mean exactly? Okay, gene therapy, this is something we're going to deal with, even though this will mainly be for tomorrow. So please do attend this conference, save the date tomorrow, gene therapy. But then cell therapy and genome editing. Today, we are mainly going to deal with two of these strategies. Leaps forward, giant leaps forward have been made in this field already, namely due to absolutely exceptional, extraordinary discoveries, so extraordinary that they have even led to a Nobel Prize, a Nobel Prize for medicine for IPS, that was in IPS cells in 2012, and another one for chemistry only a few months ago for genome editing. So for CRISPR-Cas9. So now it seems like we can start and we can have a real kickoff start with an introduction where we're going to hear about the three different strategies in terms of their enormous, huge potentialities, as I've said, but also in terms of their challenges, because these are certainly still very challenging pathways for us to tread. Then two presentations will follow about the state of the art of some preclinical trials in cell therapy and in genome editing. And both presentations will be focused on such trials to hopefully reach the clinical practice as soon as possible. The session will be concluded by a sort of a cross-cutting presentation, a very interesting one that will cover all such strategies insofar as the presentation will cover the problems connected to the use of biological products, that is the immune response. So what about our immune response to such novel products? This is something very important that, as I said, will be dealt with within the last presentation. Hopefully some solutions will also be mentioned. Actually, gene therapy, I might add, has reached the clinical practice in humans, but there are other problems, and hopefully by solving such problems, we'll manage to pave the way to such clinical trials in the human species as soon as possible. So, Annemieke Aertsma-Roos, you have the floor. We're very, very happy to have you here. Annemieke is professor of translational genomics at Leiden University. She has been dealing with DMD research for many years. She is responsible for several publications in this field, actually for 2 years in a row, namely in 2016 and 2017. She was considered as the most influential researcher in the field of DMD. So Annemieke is really a great example of how science can be communicated in a simple way, in an accessible way, of course, for scientists as well as families and everybody really, but always keeping a very high scientific rigor. Thank you very much. You have the floor, Annemieke.
Okay, thank you for the introduction, and thank you for virtually having me. I'm very sorry that we don't have an in-person meeting, and we were discussing that the Italian Parent Project meeting was the first meeting that was actually cancelled last year for me. I'm very sorry that also this year, well, at least it's not cancelled, it's there online. But I would, of course, have loved to be there in person because that is better. But online is better than nothing. So as announced, I will give an introduction in cell and gene therapy for this session, but also the session that will be there tomorrow on gene therapy. So these are my disclosures. If you want to read them, make a screenshot. And before we go into the therapeutic approaches, I'll very briefly outline what it goes wrong in Duchenne. So Duchenne patients cannot make dystrophin. And dystrophin normally stabilizes muscle fibers during contraction. If you don't have dystrophin, with every contraction, your muscle fibers are damaged. And this leads to inflammation, impaired regeneration, the formation of scar tissue, it's fibrosis and fat tissue, and eventually the loss of muscle tissue and muscle function. Now, there is also a less severe disease called Becker muscular dystrophy. These patients make partially functional dystrophin, and they have a slower disease progression. And less severe means compared to Duchenne, it's less severe, but it is, of course, still a disease. So I don't want to trivialize that. So since the cause of Duchenne is that patients cannot make dystrophin, genetic approaches are there to try and restore dystrophin. If we could bring back dystrophin into the patients, then we hope that that will be therapeutic. We can do that on DNA level, which is what I will introduce today, cell transplantation, gene therapy, genome editing. We can also do it on RNA level, exon skipping and atalurin, so stop codon read-through. That will be discussed later in the conference. I will not introduce it today. I will just focus on restoring dystrophin on DNA level. So we'll start with cell therapy. And actually, so Filippo made this joke of advanced therapies being old. Cell therapy is a very old idea. It's been there for over 40 years. And the idea is that if you isolate muscle stem cells, and you can see the muscle stem cells here on the screen. So let me get my pointer. You can see them here. So this is a muscle fiber, and you can see these blebs laying on top of the muscle fibers. Those are the muscle stem cells. So if you isolate them from a healthy donor, you culture them in the lab, you get a lot of them, and then you transplant them into a patient. You get two things. One, these muscle stem cells will repair the muscle, and two, because you isolated them from a healthy individual, these stem cells can make dystrophin. So then where they repair the muscle, also you can make dystrophin. So that is the theory behind it. But unfortunately, the reality is complicated. The first thing is, if you have these stem cells and you inject them into the bloodstream, the cells do not exit the bloodstream. They stay there and they die. So then you can say, well, maybe we should then inject them into the muscles themselves. But even if you do that, they don't migrate. And so this is a muscle biopsy stained for dystrophin. So red is dystrophin. This is before treatment. There is no red, there is no dystrophin. And this is after treatment. And what they did here, they injected a square centimeter of muscle with 250 small injections. And this was done by Jacques Tremblay in Canada. And what you can see, you see where the injection was. You see a red area here, a red area here, a red area there. But in between, there's no red area. So also within the muscles, these cells don't migrate. Now, this does not work for patients because you need so many cells. You need to inject muscles locally. You cannot do millions and millions of injections. You need too many cells, and it's just not realistic. So this approach, injecting muscle stem cells, doesn't seem to work. So are there other ways to get stem cells? Well, yes, there are. There are stem cells in the blood vessels, in fat, in bone marrow, and these cells can go from the bloodstream into the muscle and then help repair muscle. So that even though they're not from the muscle, they can still become muscle. The problem here is that if we isolate these cells, they go to the muscle with very, very low efficiency, and that is therefore not therapeutic. And then the last problem is the problem of the niche. And that is because there is no dystrophin. So there is this chronic muscle damage, there is inflammation, repair is inhibited, you get scar tissue formation. And these processes produce a lot of factors. And these factors are there very abundantly. So if you have only a few transplanted cells that reach the muscle, they will get these signals as well. So they don't receive the proper signals. They don't receive the signals they need to become muscles. They receive the signals to become scar tissue. And that is a final challenge of the cell therapy approach. Now there are other ways. And I think so what this really means is that so for a long time, we were not able to deliver cell therapy. But of course, now there are new techniques, there are new systems. So maybe the field wasn't right for it, but maybe the field will be right for it with new techniques and new approaches. And that is what you will hear in further talks. But there's also another way of cell therapy, and that is not using them to repair dystrophin, but using them to give or to produce growth factors. Because if you have stem cells from a healthy person, they will produce growth factors that tell the muscle to become muscle. So this can be beneficial for dystrophic muscle and dystrophic heart. So you can use these stem cells as a factory for growth factors. The problem is that these stem cells will die. So after a few weeks, they will all have died. So what you need, if you do this, you need to repeat the transplantation regularly. Every 3 months is currently what's being used. So this approach, so using the stem cells to produce healthy growth factors to help the muscle quality improve, this approach is currently tested in a clinical trial by a company called Capricorn, and I think they have a presentation on Sunday about this. So that is the stem cell. So I think the conclusion with stem cells is the idea has been there for a long time, but we really need newer techniques, new developments, because the challenges are big. Now, if we can't do stem cell treatment, can we then give the gene in another way? Can we, instead of giving the whole cell with the gene, can we just give the gene itself? Because the gene has the code for the protein. You can either give the whole gene, which is very big, or only the genetic code, which is called cDNA, which is much smaller. Now you need a delivery tool if you want to deliver a gene. If you just inject DNA, it will be broken down. So you need viral vectors. Viruses are very good at delivering DNA. That is what they do. Now you need a virus that can infect skeletal muscle. And there is one virus, it's called AAV, adeno-associated virus, and this can go to skeletal muscle. Most other viruses do not, but this one does. And there's different versions, so there's different subtypes, and they have numbers, and some are quite good or even better than others at going to muscles. And the ones that are really good at going to muscles are AAV9 and AAV7, and not surprisingly, these are used in clinical trials with for muscle disease patients. Now, the problem with AAV is that it has a limited capacity. So it's a very small virus, so it can only have a small piece of DNA. Well, dystrophin, as you may know, is encoded by the largest gene that we have. So we cannot fit the whole gene. We cannot fit the whole genetic code. We need to create a microdystrophin. We need to create a dystrophin that has all the crucial domains and as little of everything else. So here is a schematic depiction of dystrophin. So this is a carabiner. What dystrophin does, it binds to the structure of the infrastructure, the skeleton of the muscle fiber inside on one end, and it binds to the connective tissue layer surrounding the muscle fibers on the other side. And in between, there is this elastic domain. Now, what is crucial are these two pieces where you connect. So what you see, so this is the whole dystrophin here. And these are the 3 microdystrophins. So Sarepta, Pfizer, and Solid is doing trials with these microdystrophins. You see, they slightly vary, but they all have this domain binding to the skeleton of the muscle fiber, and this domain binding to the connective tissue. So they're really small, and they fit now in AAV. There are some outstanding questions. How functional is this microdystrophin? We know it's functional in dogs, we know it's functional in mice, but these walk on 4 legs while humans walk on 2 legs. So will it be strong enough to also deal with the force that is involved in walking on 2 legs? We don't know that yet. Another thing is we don't know how long the expression will persist. With gene therapy, you assume you have to treat once. But the problem is that AAV doesn't go inside the DNA. The AAV, the microdystrophin gene, floats around in the cells. Now, because microdystrophin is not fully functional, there will still be muscle turnover. You will still gradually lose muscle. And if you lose the muscle, you will also lose this microdystrophin. Now, how long will that take? We don't know. It could be years, but it could also be 10 years. So that is something that we'll have to wait and find out. Then, of course, the question is, if you lose it after a couple of years, can we repeat the treatment? And there the challenge is that will be difficult, because if you give someone gene therapy, you give a very high dose of AAV, of this virus, and there will be an immune response. So that is something that also currently people are working on to try and solve this challenge. Now, finally, the genome editing. So it got the Nobel Prize for chemistry, and I want to stress, it got the Nobel Prize for chemistry, not for medicine, because there's currently not a single genome editing medicine there. All the research there's a lot of research done on trying to develop this as a therapy, but it's all in early stages. So this is the schematic depiction, but I like to simplify things, as was already announced. So this is what genome editing is. It's a DNA scissor, the Cas9, and a GPS that tells the scissor where to cut in the DNA. Now, why would you want to cut in the DNA? You want to do this because then you activate the repair system. There's 2 types. There's one that works in dividing cells that can do errorless repair. So you have a mistake and you correct the mistake. And well, I use the Tipex. So you cut out the mistake and you add what is needed. Unfortunately, this works only in dividing cells, so not in muscle. So even though the system is beautiful, for Duchenne, we cannot use it. So we rely on this one, the non-homologous end joining, which I generally call the glue system. So you have this cut and you just put the 2 pieces together, and there will be some information lost because, well, at this cut, pieces of DNA will come off. However, at least the break is repaired. Now, why would you use this for Duchenne? You use this for Duchenne to achieve permanent exon skipping. I'm going to very briefly explain what exon skipping is, because it's one of those RNA techniques, so that targets the transcripts. And what we know, so why can't Duchenne patients make dystrophin? Because the code is unreadable. And you see here an example. So the code is located on exons. You see here exons. You see here also 52 is missing. And you see that the code fits. So this fits one. This one fits to this one. This one fits to this one. But 51 doesn't fit to 53. So the code is disrupted. The code becomes unreadable. This patient cannot make dystrophin. Now, what exon skipping tries to do is try to restore the code. And you can see 50 would fit to 53. So if we could somehow make exon 51 go away, the code would be restored, and then the code becomes readable, and you can make a dystrophin like Becker patients make. Now, normally with exon skipping, you do this on the transcript. So you have the DNA, a temporary copy is made, RNA, and that is then translated into protein. That means you have to repeat treatment because this is a temporary copy. So after a few days, it's gone, and you need to do this again. So you need to repeat the treatment. Currently, the approved drug in the US, you have to repeat the treatment every week, intravenous infusion. So if you could cut on the DNA level, then you would have to do this only once, because every time you make a copy from this, if this 51 is cut out by the scissor, every time you get one that is readable. So that is the advantage of CRISPR genome editing over exon skipping. You would have to do it only once. Now, people are very enthusiastic about genome editing. And that makes sense because the technique has a lot of potential. For the first time, we can make changes in DNA in a very targeted way. And we can generate disease models in cells, in animals, very easy, very quickly. What normally would take years now takes less than a year. So that is, of course, great. And that is why also the Nobel Prize for Chemistry was awarded. Of course, then you also think, well, then it can have therapeutic options. And that is in early stages yet. It's easy in cultured cells. It's easy, well, it works in animal models. It has had a lot of media attention, but there are still challenges. So the first one is the delivery. You need to deliver the GPS system and the scissor to all the muscles. For this, you need to use AAV. You need to use the viral vector. Now, for microdystrophin, for adding this gene, this works. But here you have a two-component system because you need one AAV for the scissor and one for the GPS. And it's also a two-step process. There's no guarantee that if you deliver the system, that there will actually be a cut. So that makes it less efficient than just adding the gene and having the protein. The efficiency is currently low. And then, of course, the biggest question is, how specific are these scissors? Do they only cut where they have to cut or also somewhere else? Well, we know at the moment they're also cutting in other locations. And that is, of course, not what we want, because if there is a change in the DNA, we cannot undo that. So people who say CRISPR cure, I have to disappoint. And I know that people get very enthusiastic and hopeful about genome editing. Unfortunately, there's no such thing as a CRISPR cure for Duchenne, because what you do is you restore the code. The code becomes readable and you make a Becker-like protein that's only partially functional. So it is not a cure. It does not halt muscular dystrophy. It hopefully slows it down. And also it depends on the time of intervention, and that goes for all different therapies, unfortunately. We have no way yet to restore the muscle that is lost and the muscle function that is lost. So it's important to treat early, not just with genome editing, which is in preclinical stage, but with all the different therapy. So recapping, there's cell transplantation. It's been tested in trials so far for dystrophin restoration. So far that has failed, but there's now other approaches to try and improve muscle quality. There's the gene therapy that's also in trials. You will hear more about that tomorrow. And then there's the genome editing, which is in preclinical stages, so only in model systems as yet. And that was my last slide. So thank you very much for your attention. And I give the floor back to Filippo.
Thank you. Grazie Annemiek. Allora, approfittiamo per ringraziare
Thank you very much, Annemiek. We wish to thank you for being so exceptionally clear. Thank you very much indeed. Now, having said that, despite Annemieke being so exceptionally clear, in case you have some questions, please use the chat on Zoom. Just write down your questions. You can do it now if you so wish. And at the end of the session, we'll be reading them out to our outstanding presenters. So they'll try to answer your questions. Annemieke, thank you again, but of course you'll still be with us for the Q&A session. Now the floor goes to Rita Berlinguero. She's a professor of medicine at the University of Minnesota. Well, I actually met her in 2012, I believe, because she came, I think, to Rome. She had produced a great paper. At that time, she published it on IPS, on an important journal. And because we've always been so very curious about whatever happens in the scientific world, we came into touch with her and invited her to come to Rome. Today, she's with us again, precisely to let us know about what has happened in this field in the meantime. Maybe there's a clinical practice soon out there for us, an advanced therapy, actually. Thank you very much, Rita. You have the floor.
Thank you. Thank you, the organizers, for the invitation. It's always a huge pleasure to speak to this community. I had the privilege to present in the parent project in Milan back in 2008, in the very beginning of our work. We are really the first proof of principle with mouse cells. The second meeting in Rome, we evolved to human cells. We are progressing slowly. And I'm very happy to discuss about our path from the laboratory, from the bench to the bedside. It's a long road. There are lots of challenges, as previously discussed in the previous talk, but we have to try. That's how we feel it. We have to face the challenges as the patients face the challenges every day. So we need to try at least and see if you can circumvent the challenges. So I prefer to be in Italy right now. I'm in cold Minnesota, but I'm happy to share our data and where we are by Zoom. So just to briefly discuss, there are generally two modalities of cell therapy, and they both have advantages and disadvantages. When we are talking about allogeneic, you have a donor, a healthy donor, especially when you're talking about genetic diseases, and the cells from this donor can be used for a large number of patients, okay? So basically off the shelf. Of course, this is a safe, cheaper strategy, and it can be easier. Of course, it has more challenges in terms of the immune system. In the autologous setting, the patient is the donor. So the cells are isolated from the patients, may be manipulated ex vivo in the dish and put it back into the patient. So this has advantages in terms of the immune system in the same, and is specific to the patient. It's just more expensive and a longer journey. But these two strategies are possible. So why IPS cells? What are induced pluripotent cells? The previous speaker talked about muscle stem cells and the challenges involved. I also talk about other cell types isolated from the body. So these are different, and as alluded in the introduction, that was Yamanaka received the Nobel Prize for Medicine, is the potential. Still, we are talking about the potential. We are not in clinical trials for muscular dystrophies yet, but I say IPS cell-derived cell types are currently in clinical trials for macular degeneration and Parkinson's disease. Muscular dystrophies are more challenging. Duchenne is more challenging considering the delivery, but we have to try to circumvent this issue. So what are induced pluripotent stem cells? These are cells reprogrammed from any adult tissue. And why we are interested? Why do you think these cells might be different from other cell types that have been studied for a long time. Well, first is that you can generate very large numbers of the cells in the dish, what you cannot obtain with other cell types. Second is that these cells have the potential to generate any cell type in the body, specifically muscle for our interest, which we have developed the technology for it. And third is that they are amenable for genetic modification. So you're focusing on the autologous cell transplantation. We obviously need to provide the gene correction of the dystrophin gene. Not easy, but it's possible. Translating all this to the clinic is another challenge, but all those different components are possible. So this is just a cartoon to show briefly how this works. So if you have a healthy donor in the context of an allogenic setting, you get skin or blood cells because it's just the easiest material to obtain. You reprogram with a cocktail of factors, which was the discovery that we had the Nobel Prize for iPS cells. Once we have iPS cells from this healthy donor, we can differentiate those cells in myogenic progenitors and generate transplantable myogenic cells that theoretically can be used in an allogenics for a DMD patient. The autologous strategy, you generate these iPS cells from the patient, you reprogram, and you still have the iPS cells with mutations. So here we needed to add the important step, that's the gene correction, that we can use CRISPR-Cas9 and do the myogenic differentiation. Now you have transplantable gene correction, gene-corrected iPS cell myogenic progenitors that could theoretically be used in the patient. So what is unique to what we have been doing for the last 15 years is this strategy of actually generating myogenic progenitors, not a more differentiated cell myoblast, but a cell that once you transplant can make muscle fibers and muscle satellite cells. Up to now in mice, these are our patients to date because we have to start there and hopefully we can evolve. I'll tell you where we are right now. So we have in the last 15 years show extensive proof of concept for both strategies, autologous, allogeneic and autologous in terms of performing gene correction of iPS cells from patients and showing validation in the dish and into animal models of muscular dystrophy. That's all proof of concept. So very brief, why this is important, why do you think this might be different from previous studies? Well, again, as I mentioned, the dish, using our conditional expression of these important transcription factors, it really enables us to generate this cell type here, that's really what you want, very early myogenic progenitors or proliferating cells that once you transplant are able to make muscle in the mouse and also make muscle stem cells. It's the cells that are localized in this area here, adjacent to the cell. And we have shown that these cells transplanting the muscle, intramuscular or systemic delivery, they actually do migrate, are able to improve muscle function. So very important point is that they are able to make, at least in mice, they are able to make muscle fibers and distribute within the muscles. I have better images to show in a little bit. They make muscle stem cells and improve function. Again, keep this optimism with a grain of salt because this is not the patient and doesn't have all the levels of complexity. Mice are very simple, but we have to start there. Otherwise, we will never advance. So if there is proof of concept in the mice, we feel hopeful. Doesn't mean that we don't have many, many challenges ahead of us. So what's the roadmap? So after we have done all this proof of concept, we were like, well, if you want to test if this is a possibility, where do you start? And this is a very summarized map. And for a long time, we are here, right? We had the proof of concept, but how do you get this proof of concept to a first in human clinical trial? The list is just enormous, and it's definitely not something that one can do alone. So what I did back in January of 2016, about 5 years ago, we put together in the University of Minnesota a translational working group to enable the potential, I would say potential therapeutic application of the cell. So getting from the beginning, what do you need? From the manufacturing point of view to all the preclinical studies and FDA. So we have people here with different expertises in cell therapy, clinical grade production. We have this facility in Minnesota, experienced FDA and neurologists, of course. So the list is long, and I love to-do lists, but what I really love about to-do lists is mark them down. So in dark blue here, you can see all the things we have done in the last 5 years. So we study deeply methodology to generate cell product. This is the best we can do. Very importantly, when using iPS cells is the purification of the cell product. Make sure that the cells you are generating are compliant, so they actually can be used in patients, because a lot of reagents sometimes used in the lab are not. So we eliminated all the components that wouldn't be compatible with a clinical trial. Of course, you have to make sure that you actually can generate very large numbers. We are able to do in the lab, but can you do this in a compliant method that can be used in patients? And of course, you have to have characterization of the product. And then where we are now is transferring. We were able to show all these points. Where we are doing now is actually finalizing the transfer of this technology we have been doing every day in my laboratory to this clinical grade facility, which is doing cell production for transplantation to patients. That's their job. So iPS is a new thing for them, and in particular, our manufacture of muscle cells. So we have to make sure that all the paperwork, everything is validated and done by this facility. And you need all clinical grade reagents. So once you are there, then you are ready to have the clinical product and test preclinically, and hopefully if everything goes well, IND filing to start a safety trial. So just to show you, we started with clinical grade iPS cells generated by NIH specifically for translational applications. And we want to make sure that we actually can validate the cells using our technology. So these are how the induced pluripotent stem cells look like. They are normal in terms of karyotype. The cells make nice myotubes in the dish. And once you transplant them in this dystrophin deficient mice, we can see nice donor-derived muscle. I'm not gonna bore you. I know I have limited time, so I just wanna give you a sense of all the optimization, lots and lots of kind of boring work, just eliminating different components and seeing what would be the ideal culture conditions. So we came up with a media composition that reduces significantly serum, and you are able to see persistent differentiation and engraftment. Optimization of purification. So it's very important not to use cells that are homogeneous, so eliminate any non-muscle cell that you might have in your cultures. So we use this particular antibody to purify the cells that we have validated. And you did a lot of optimization in terms of antibody concentration to make sure that everything is optimal. And you came up with this plan that you presented to the FDA, which you have clinical grade iPS cells. You change those cells with this PAX7 because this is a very important point to actually specifically instruct the cells to make skeletal muscle. At this point, they are just modified, but they are present with PAX7. We have a PAX7 master cell bank, so lots and lots of vials in the freezer. At this point, we do a whole genome sequence and analyzing the cells to see if they're safe. And then you generate the product. If everything goes well, you generated the myogenic cell product. That's this cell here in the end after the purification. So can you really generate large numbers? So one key aspect, as I mentioned, is the purification combined with this machine you are seeing here that's called Clinimax. Because Clinimax is a way of purifying the cells, but as you can see there, the cells goes in this bag. So it's a
Rita, well,
yeah.
we need to close.
Okay, so this is possible. So I just want to show you can generate very large numbers of cells here. And off the shelf, we are able to thaw the cells and inject directly into the mice, and you see lots of muscle. And so validating the idea off the shelf. So that's where we are. We move it from here to here, and hopefully within the next 1-2 years, we'll be here if everything keeps moving forward. And I'll just move along, and thank you all for your attention. I'll be happy to answer questions in the end.
Jairo per questa presentazione. È interessante vedere come queste strategie lo sviluppo di queste strategie.
Thank you for this presentation, and we see that there are genome editing, and then the vector that is used is the same as for gene therapy. So it is very important for everything to move forward in all directions. So we need a cultivated field for a plant to grow. And now we give the floor to Eleonora Maino. She is a very young Italian researcher who works with the group of Ronald Cold at the Hospital for Sick Children in Toronto. The CON group has been working for many years with the idea of creating revolutionary treatment for genetic diseases, special attention for DMD. Over the last years, they concentrated on CRISPR-Cas9 based therapies, so genome editing. And now I give you the floor for 10 minutes, and please don't speak too fast for translation. Good afternoon. Thank you for the introduction. It is a great pleasure for me to be here and speak to an Italian audience about my PhD work. Today, as Ilaria was telling you, I'll be speaking about all those therapies that are called genome editing, so RNA modification, and specifically about DMD. Anime introduction is very useful because, of course, the focal center of this talk is this CRISPR-Cas9, is this technology of genome editing that was discovered in 2012, that has been changing the way in which we do research and also the way in which we think about new therapies for genetic diseases such as DMD. As you have seen, CRISPR-Cas9 has a very simple system. It is formed by two main components. Cas9, that is this protein that works as a sort of scissors, and then the RNA guide, that is the GPS, so to speak, that addresses Cas9 to a specific position in DNA. When it is there, it can cut DNA, and we can exploit this system to develop therapeutic strategies for a number of genetic diseases. Let's see how it was used for DMD. To do that, we must sort of make a step backwards, go back to the genetics of Duchenne. It is caused by mutation in the dystrophin gene, and something very typical of this disease that is that there are different mutations that may cause it. 70% of patients lack a region of the gene, so it is a deletion. 20% have point mutation, so and 10% of patients have a duplications of some parts of the gene. The result of all these changes leads to the absence of the expression of dystrophin, and so the presence of muscle damage and all the symptoms of the disease. Up till now, we have developed therapies essentially therapies for point mutation. As Annika said at the beginning, we can use approaches that are called Axon Skipping approaches, and permanent exon skipping therapies can be defined as genome editing. We can modify DNA so as to create shorter forms but working forms of this protein. 3 papers were published in Genome Editing in Duchenne. They were published 5 years ago, and they opened up the way for the development of this type of therapeutic strategies. And now I'd like to make an example to this, give you an idea of how CRISPR-Cas9 can be used for this type of therapeutical approaches. So we may have deletion mutation. Here there is this part missing, 50, or point mutation, where we have a mutated region. What we can do is either removing another small bit or removing the mutated region to restore the expression of a shorter but working form of the dystrophin gene. And we can do this with our system, CRISPR-Cas9. So we can identify, well, they can be located in two positions, before and after the area to be eliminated. And after the cut, you have the elimination of that area, and you can create this shorter but functional forms of the gene. Other approaches have been considered and tested in other animals, as dogs or pigs. And we hope that soon these strategies can really move further. What I work on, I mean, my PhD is on this 10% of patients, duplications of the gene causing the disease. So in this second part of my talk, I'd like to tell you how we can use CRISPR-Cas9 to develop therapies for DMD patients having duplications mutations. So what we did is, having this animal model, having this area that is repeated twice. This mimics a mutation that has been found in a patient. This animal model develops symptoms that are typical of the disease, and then we try to develop a genome editing strategy with CRISPR-Cas9 to correct the mutation and improve the health and symptoms of muscle dystrophy in this animal. And so we start trying to understand how we can really modify this gene to the sgRNA to solve this problem. There is something typical. We have the same sequence that is repeated twice, and the sequence is absolutely identical. So what we can do is designing just one RNA guide, so just one GPS guide, and bringing our scissor to some regions exactly at the same spot. So we can eliminate this extra bit of DNA and reconstruct a dystrophin gene that is perfectly functional, a gene that has all the domains present. And this is what we have tried to do, use this strategy in vivo in our model. That's what we did. We took our therapeutical components and we packed them in these viral vectors. You have already heard about these. And we have injected them in mice, in newborn mice, and we checked the effect of treatment 7 weeks later. Obviously, we want to look at the effect of treatment essentially in the muscle, because the muscle is the organ that is more affected by the disease. You see that in healthy mice, dystrophin is expressed in all muscle fibers, and the muscle is very healthy. When we have mice with a duplication, we have no dystrophin expression, and there is muscle damage. In our mice that were treated with Cas9, you can see that we restore the expression of dystrophin, and this leads to a general improvement in muscle health. But we also want to see whether the muscle is functional, and to do that, we used a test checking how animals move in a room, in a box. In black, we have healthy mice. Mice with a duplication have a very limited mobility, whereas the mice that were that received the treatment show an improvement of symptoms. They start exploring the environment, and they have levels of activity that are similar to those of a normal mice. So Cas9, I mean, this system can be used for duplication mutations in DMD, and it has the potential of being used in different types of genetic diseases. But there is a but, and that is what Nike told. It is a very young discovery. It was discovered 9 years ago, so there are still many questions to be answered before getting to the clinical level. First of all, the specificity of the system. We know that Cas9 can create off targets. It can make small mistakes in sites that are very similar to its main site, to its main goal. Viral vectors and Cas9, of course, as well, can stimulate an immune response once they administered. So it is also important to understand whether we can develop other systems such as nanoparticles that could be better tolerated during treatment. And then, as for all diseases, more specifically for DMD, we must truly understand when we can administer our treatment for the therapy to work just fine. I wish to thank my lab, my supervisors, and all the members of the lab who have truly worked hard with me on the project, our co-workers, our funders, and all of you for your attention. And of course, if there is any question, all questions are welcome. Thank you.
E ti chiediamo appunto di restare con noi.
Thank you. Thanks a lot. Please stay with us for the Q&A session. And now the floor goes to Barry Bernish, the director of the Center for Gene Therapy of the University of Florida, and Manuela Corti, who's working with him. Well, Barry, are you an old friend? That doesn't sound that nice, does it? So we've been long-lasting friends, let's put it that way. We've been cooperating also with Professor Irene Bozzoni. Since then, we were trying to find a solution for using AAVs, and therefore we have held a cooperation with Barry Byrne for some time. And I mean, the reason why I'm saying this is that Professor Byrne has been working for years about this subject, which is of paramount importance. Now, Byrne's and Corti's presentation will be focused on something very important that is cross-cutting, if you wish, namely the immune response. Indeed, today, the immune response is, you know, a key phrase in our world, in our field. But I mean, this is just to say that it is really a very important issue, because it's always fundamental, it's always crucial. So please, Barry Byrne and Manuela Corti, keep the time schedule of 15 minutes because we are running a bit late already. So having said that, please, you have the floor. Thank you.
Thank you very much. Can you hear me okay? Yes? Okay, I'll proceed then. Thank you. Thank you again for including us in this great meeting. We wish we could be there in person and hopefully next year. So I'm going to talk about some of the work we've done in the form of a series of questions and answers so that we can really make this most understandable to the audience and really have a great level of participation. So hopefully this will be helpful also for the sessions that are to follow tomorrow. We heard a little bit about AAV. I think it's always helpful to just see what AAV actually is. This model represents AAV, and actually I realize that I have also it right behind me on the shelf here. So this is an AAV vector in a very enlarged form from a 3D map that one of our faculty helped generate here at the University of Florida. There are many forms of AAV exist in nature, and this is one of the issues that we face in understanding what the natural immunity is to AAV because of the exposure. Now, in the natural state, it really is unique in that it does not cause any known disease, but in the context of a therapeutic use, we found that the immune responses are a significant barrier to overcome. So I thought I'd play this brief video to show conceptually how therapeutic genes are put inside of AAV with a control element called a promoter. The functional gene is contained within packaging signals for AAV. When that is put inside of that structure that I just showed you, it's able to leave the circulation and go into a cell. So this is kind of a depiction of how AAV would be delivered through, in the case of muscular dystrophy, through the bloodstream to reach all the muscle cells, or at least as many as possible, travel to where all the controls for gene expression are made in the nucleus. And this provides the instructions on how to make the gene that's carried by the AAV vector, or in the case of CRISPR, as we heard, is the means for delivering the CRISPR editing machinery and the guide RNAs into the cell. And this persists for a long time, particularly in cells that don't divide any further, really essentially the life of those cells. But when we are exposed to this for the first time, there are responses like to any viral infection or an immunization for that matter. And so I'm going to say a little bit about, now that we know that AAV is composed of DNA and a protein, that leads to an immune response, both an early, an intense immune response that makes, in fact, as many in the community have heard, even in placebo-controlled trials, is quite evident to those that get the AAV vector and not the control or the placebo, that there's a brief illness that follows the exposure. And this is the early immune response known as innate immunity. So the innate immune response can facilitate both the production of antibodies and augment the adaptive immune response, which can have a cellular component or lead to antibody formation. So these antibodies are a key issue, both because they influence the eligibility to receive the therapy and they influence safety findings afterwards. So this is a cartoon or depiction of what an antibody is. It's a protein that's stable in the blood. We acquire antibodies through environmental exposures, or in an infant, they're passively transferred from the mother, both in utero and through breast milk. The antibodies bind to antigens. In this case, the antigen would be AAV. That limits the effectiveness of the AAV therapy or prevents it from working in the first place. When you're exposed to a new protein for the first time, there's a certain type of immunoglobulin made called IgM. Later, after the exposure, there's a switch to this more stable, prominent form of immunoglobulin that lasts in the serum for a long time. And if you get exposed a second time, the IgM response is lesser than the IgG response. So we can tell whether patients have been seeing something for the first time or they were previously exposed. And unfortunately, about half of adult and adolescent patients, particularly in Duchenne, in a population we've surveyed, have high IgG antibodies against AAV. So we've been very focused on ways to lessen this response and or prevent it from causing adverse events in the studies or clinically. It's important to say a few things about the kinds of assays that are used. I'm going to jump through this quickly just to say that, one, you sometimes hear about a neutralizing antibody assay. These are done in tissue culture against cells. They are hard to perform, in fact, hard to do reproducibly. We rely, and some of the data we'll show is based on this antibody binding assay called an ELISA assay, called Enzyme-Linked Immunoabsorbent assay. This is done against a specific serotype of AAV, but there's many cross-reactive antibodies. And these antibodies respond to all parts of that viral protein. This is very reproducible. It's what's being used commercially now for access to the commercial product called Zolgensma. And it works at very low dilution that lets us predict whether someone has been previously exposed. And so one of the things we wanted to address is, since a high proportion of individuals may have prior AAV exposure in nature, we want, and that excludes them from trials, we wanted to do studies to understand, is the threshold used in clinical trials, which in this case is this dotted line, is that appropriate in all cases? So this patient in the green, for example, would be eligible to receive AAV right away. But what if you were this person who was above the threshold at one time, but then subsequently became eligible? That might be an easily accepted recipient of AAV. But this person who has a very high titer that never goes down, we'd have to imagine ways to make them eligible. And we have studied some immune modulation regimens to decrease pre-existing levels of AAV and to determine what in fact precludes treatment. And Manuel is going to pick up at this point and talk about how this is relevant to DMD and the durability of the therapy. So I'll stop sharing and turn it over to her. I hope I'll stop sharing.
Manuela.
Ora continua Manuela, immagino.
Yes, can you hear me?
Yes.
Sì.
Yes. Good afternoon, everyone. Thank you for Dr. Berndt for introducing the immune responses to AAV. In the next slide, I will address other questions relating to these immune responses to AAV, but most importantly, a strategy to overcome these immune responses. So the question is a gene therapy for DMD durable for the lifespan? And unfortunately, the answer is probably not. And especially because we are dealing with muscle and regeneration of muscle. And how this affects the treatment is that early exposure to gene therapy leads to a less durable treatment. But let's try to understand why it's less durable. The reason is because myoblasts, that are the progenitor cell for muscle cell, are not exposed to AAV, and therefore new muscle that are not present at time of gene therapy dosing will not present gene therapy. And this, we demonstrate this in an experiment in primates. In this experiment, we injected primates at birth with gene therapy, and we look at the transgene activity through muscle biopsy throughout the first year. As you can see, the transgene activity reduced throughout the year, almost getting to the level of 0 after a year. And this is what we expect will happen in a growing child due to growth and new muscle. So thanks to Dr. Berndt that introduced how this immunoresponse work, we recognize that immunoresponse to AAV is a universal problem to all gene therapy. And the reason is because there is a primary antibody formation that affect the clearance of the vector in the circulation, and so reduce the efficacy, but also preclude the repeat dosing of AAV. But in our group S study, for the last 5 years, a strategy to overcome these immunoresponses so that we could allow for repeat dosing. I'm not going into details in this complicated graph, but basically this is just to show you that when the vector, the gene therapy is introduced in our body, is recognized by the antigen presenting cell, which activate the cell that are at the basic of the immunoresponses, the T cell pathway and the B cell pathway. The B cell produce the antibody that Dr. Berndt was talking about. So we use 2 drugs that are immunomodulation drugs that basically, the first one is rituximab, that is an antibody that attached to the CD20 protein present on the B cell, and basically induce a depletion of the B cell. Therefore, we will not have antibody against the B cell. And also we use sirolimus that is activating the T cell pathway and also modulating the B cell. So the combination of rituximab and sirolimus prevent the formation of an antibody and allow for repeat dosing. Here we use this strategy in several clinical trials. Here we are, I'm showing one clinical trial where we dosed the individual twice with AAV. So we perform an immunosuppression regimen before the AAV exposure. Then the individual receive the first AAV and 4 months later receive a second AAV exposure. What I was showing here is the antibody, IgG and IgM in this individual that received immunomodulation remain low throughout the study. That is not what happened in those individuals that did not receive the immunomodulation. So we were successfully we were able to block the antibody against the AAV. So why this is relevant to DMD? As we said, the immunoresponse against the capsid is a general problem, so it's important to prevent this antibody. But also it's important because it can preclude, as Dr. Brown mentioned, the participation of lots of individuals in the clinical trial, and this is relevant for older patients. But also enable early treatment, especially with the advent of newborn screening. So early treatment, we will be able to redose later in the lifespan using this immunomodulation. So because it's relevant to a DMD, we run some experiment in the DMDX mice. So we use the vector carrying the Microdystrophin. As you can see here in this graph, we are showing animal that received the vector twice. As you can see, if the immunomodulation was not used, the antibody are super high. But when we use the immunomodulation in combination with the vector, we were able to block the antibody. So we were successful to prevent the immunoresponses. Another question important is, can a 16 anti-AAV antibody be reduced and allow for those individuals that have already been exposed to AAV participate to clinical trial? And the answer is yes. We performed some experiment in mice where we expose the animal to AAV and we create a pre-immunity. And then we test their antibody titer before starting an immunosuppression regimen. Actually, we compare 4 different immunosuppression regimens. And then we test the antibody again. What we observed is that we were able to reduce the antibody titer against the AAV with all the regimen the immunosuppression regimen we will use compared to the pre-immune animal. So this is still these are still pilot studies in mice, but we are about to start also some primate work. And this work is being supported by Duchenne UK Foundation. That's why it's important that the steroids that are considered immunosuppression are not really affecting the antibody titer. So the steroids are useful to manage an inflammation, but not the antibody against the AAV. So the take-home message for this presentation is that gene replacement using AAV is a successful strategy. But we also need to manage the immunoresponses to AAV, which is a universal problem to all gene therapy. We have been successful to block this antibody against the AAV using an immunomodulation that is based on the use of rituximab and sirolimus. And these allow for repeat dosing of AAV, and that allow also for early exposure and early treatment with the advent of newborn screening. It's also important to underline that immunomodulation can be also used to manage pre-immunity so that all individuals, even with a high titer of antibody, can participate to trials. And we have to recognize that there are some side effects with gene therapy, but this can be managed with the use of immunomodulation, and we always need to understand the ratio between risk and benefits in this trial. So this is our team. We really need to thank all the patients and the participants that offered their time to our study and all our funding, including the Duchenne UK Foundation. Thank you.
Grazie. E soprattutto grazie per la precisione.
Thank you, and thank you also for the precision, for respecting time limits, even though we know it is difficult with these topics. Thank you. Thank you. All the speakers were really perfect. I mean, having a sort of TV timing is not easy for a scientific conference. Now, some questions. Well, first question, Annemieke tried to answer, but questions are visible only to speakers. So, Annemieke, could you please repeat your answer to the first question? What treatments are available for deletions of exon 45? Could you please repeat the answer? Because
yes, of course. So for exon 45 deletion, if you want to make the code readable again, you would need exon 44 skipping, or if you use genome editing, you need to cut out exon 44. But it's good to bear in mind that for microdystrophin therapy or for many other therapies, cell therapy, for instance, it's not always mutation specific. So microdystrophin should work for all mutations. And a lot of the other therapies that are in development that don't restore dystrophin also would apply to all mutations. So what is currently available for an exon 45 deletion? Nothing, unfortunately. For mutation-specific ones, it would be exon 45 skipping or exon 45 genome editing, not yet available. And then also the microdystrophin, the cell therapy, all the other ones that you've heard about today or will hear about later are all in development. And if people don't mention that it's mutation-specific, it would apply to all mutations.
Grazie Annemieke.
Grazie Annemieke. Facciamo adesso un'altra domanda che è stata già...
Another question that was asked yesterday, but I think the opinion of other experts is also very useful because it's a truly a hot topic for families. Christopher asks, what is your opinion about mesenchymal stem cells coming from the umbilical cord or the placenta, and if you are aware of clinical studies on this potential treatment? I don't know who wishes to answer. Rita, maybe? Rita?
Sure. Yeah, thanks for the question. So I don't think there is much work on specifically this source of mesenchymal stem cells, but in general, some studies have been previously performed, and the capacity of the cells to regenerate skeletal muscle is very limited. So it goes to one of those issues discussed in the first talk. One idea is, can you reprogram those mesenchymal cells into iPS and making skeletal muscle? But that's a long detour. Not sure if there is any advantage in using a cell from the placenta versus a cell, a blood cell, for example. So maybe some other speaker has more information on that, but from my knowledge, there is not much of that. It's limited regeneration for muscle.
No, it's the problem that I introduced and that Rita is now reiterating. There's no reason why these umbilical cells would somehow be better than other cells, and well, so yeah, they have no special advantage, unfortunately. So for now, they're not therapeutic. More work is needed.
Okay. Abbiamo un'altra domanda.
We have another question by Costantino. Gene therapy doesn't involve neoblasts. So once the adenovirus effect is over, the correction is no longer present. So again, the muscle goes back to being diseased. Have I understood correctly?
Yes, the myoblasts are unique in that they stay very quiet in a unique location, so-called quiescent cells in the under the basal lamina where there's not a great exposure to the gene therapy vector. So when they begin to divide with muscle damage, they don't carry the gene therapy vector into the muscle, and only the number of copies that were there from the time of dosing are retained. And over a period of time, that will ultimately lessen the expression of dystrophin in the whole fiber. So some parts may retain expression, and other areas where the myoblasts that carry the mutation have entered will not be protected from damage. So it's unknown how long that it will last, but certainly longer than in an active cell that's actively dividing, like from the blood system. But it will mean that over the lifespan, for those that are treated very early, there's likely a need to focus either on editing in the myoblast, which would be tremendous if that were able to be targeted, because then those healthy cells would populate all of the muscle over time. And then in lieu of that, as that's being developed, this concept of being able to sustain the expression through repeat exposures is a consideration we're pursuing. This is also relevant to adult patients, because young adults and adults who have 2 or 3 times the body size have a much higher exposure to the vector, and the immune responses in that scenario have been associated with some safety signals that are concerning, especially for those patients that may have fragile heart function. We don't want those problems to occur. So blocking the immune responses and or the other adaptive responses is critical to safety as well.
Grazie Barry per questa risposta molto esaustiva.
Thank you for this very exhaustive answer. And now a new question by Alessandro, asking if we can have engineered vectors that can transfer the whole dystrophin gene. Who wants to answer?
Well, what I can say is that we made a mouse model that has the whole human gene. So not just the genetic code, but the whole gene. And that was very complicated. And we could do it because we could treat the embryonic stem cells. So the one cell organism, we could manipulate those, and then we got that model. Doing that in, say, a baby that's already born, or even an older person, the whole gene, that is, it is not possible, because the gene is way too large. So it would not be possible. It may be possible in cells outside of the body, but it would still be extremely difficult to do, because it is huge. It is about 30 times bigger than the average gene.
Ok. E abbiamo un'altra domanda di
We have another question by Alberto, who is asking if we know something about the ImmTOR platform by Selecta that would enable multiple modular administration of gene therapy. The clinical study has started for AV8, and he asks whether you have any information about this method.
Yeah, ImmTOR is a drug provided by Selecta, which complexes rapamycin, which we spoke about. In our studies, this is taken orally in the ImmTOR program. It's delivered along with the gene therapy agent into the bloodstream to try to increase the level of rapamycin, particularly in the liver. So the approach has been focused mostly on liver-directed gene therapy. This may be beneficial for muscle gene therapy, but it's not known at this point.
Barry di nuovo, e c'è un'altra domanda da parte di Giulio.
Barry again, and then there is a question for Giulio to Bellingeiro to know if cells can produce functional dystrophin in adequate quantities to make a difference.
Yeah, luckily that has been reasonably well established by the first Sarepta study, as well as some of the information received from the Pfizer program, and then at the Muscular Dystrophy Association meeting coming up next month, and as well as from Dr. Morris, you'll hear tomorrow, I think, some aspects of what's been done in the solid bio study. So these are all, I think, leading to favorable outcomes related to level of dystrophin expected to be clinically meaningful. It's challenging when they're very young subjects in the study who are gaining skills, and to differentiate them from the controls who are also gaining skills. No studies other than the one by Sal that have been done in non-ambulant patients yet. This is another interesting topic for the community, since there's so much representation of those later age boys and young men. So that remains to be seen in the older population.
Anche Rita Perlingeiro vuole aggiungere su questo.
Rita Forlingeiro, would you like to add something?
Yes. Thanks for the question. Actually, I can clarify how you are starting this process. I talk about autologous and allogeneic, right? So from the patient in correction and putting back versus starting with unaffected cells. That's the way we are beginning this this preclinical work, or these attempts for a first-in-human trial, because it will allow us to optimize all the manufacturing and test safety in a number of patients. So the cells that are generated are coming from an unaffected iPS cell and generating unaffected myogenic progenitors, meaning they express normal levels of dystrophin, and you see normal expression in vivo and functional recovery. So if you are working on autologous cells, then indeed you need to make sure that you are rescuing normal levels of dystrophin. So we are doing this in the lab right now, but not at the preclinical stage yet. Did I answer the question?
Grazie Rita.
Thank you. We have another question by Alessandro asking us, what can you tell us about the Arcus platform of Lilly Pharmaceuticals? Our families have studied a lot. Arcus platform. Well, we'll organize a webinar with Lilly to understand whether they can answer the question, unless some of you wishes to give an answer. We have another question. If a patient has participated in the screening for a trial of gene therapy and has been excluded because he has shown a positivity to antibodies, is he automatically excluded also from other future trials of gene therapy if he was positive to antibodies?
I don't know, Barry, if I can start and try to take it. So it will depend from the threshold that that specific study uses. Those thresholds are I don't want to say artificial, but they are not. We really need to do more work understanding what is that threshold antibody to establish if that threshold can be bring a little bit higher so that other individuals can participate to studies. So it is not automatically excluded. It will probably be tested for other trial. But with the more and more work that we are doing and others, a better understanding of that threshold and moving that threshold a little bit higher, we will be able to include more individual. But also, as we mentioned in the presentation, we are also working on strategy to reduce that antibody below that threshold that is used for that specific trials. So I don't know if I answered your question or if Barry has more to add.
No, it's very individualized. The thresholds that are established are essentially very low at almost the lower limit of the assay. So only through testing progressively higher levels in some individuals will this be enabled to expand. And we've recently studied this in a subject with SMA who, because of newborn screening and maternal antibody, had a very high antibody titer at the time of diagnosis. It in fact took 10 months for that titer to lessen naturally. Maybe this is not such a long time in the Duchenne community, but in SMA, that without treatment, without an alternative treatment, that would have rendered that person ineligible altogether because of progression of the disease. So we do think it's really needed area to try to attack this problem, to aggressively lower the thresholds, enable participation in the studies, and determine whether there really are safety considerations or is it really just how much that impacts the efficacy. So we're trying to get there.
And maybe one thing to add is that for Duchenne, there's now 3 different microdystrophin gene therapy trials. One uses AAV7 and 2 others use AAV9. Now, if you have antibodies against AAV9, you may still be okay to receive AAV7 if there's no cross-reactivity. So then they would have to test whether you're positive for AAV7 as well or not. And vice versa, if you have antibodies for AAV7, you might still be eligible to receive AAV9. But that is something to bear in mind, I think.
Chiaro.
Grazie. Abbiamo una domanda complessa perché...
So now we have quite a difficult question because hopefully it goes with hope and hopes are always difficult to be measured. Can we have the same hopes for duplication as we do for deletions with gene therapy? Well, maybe I can try to answer. So regarding gene therapies, when we talk about, for example, microdystrophin trials, well, of course, they do not depend on mutations. So the same approach may indeed be applicable, you know, for patients having different kinds of mutations, which may include both deletions and duplications. As for genome editing strategies, mainly focused on DNA, well, we are in very initial stages, you know, even within the preclinical framework, but duplications seem to have quite a wide-ranging potentiality. You know, I'm thinking of what we are currently doing in our lab. So the gene can be reconstructed in all its functions. There's no domain lacking. So maybe in the future, this will also be the case for deletions, where possibly several therapeutical windows may be opened. But of course, there are still many questions to be answered. Thank you. Well, there may be just the last question because we have 4 minutes left and then the parallel sessions will have to take over. So the last question, at least in my opinion, may need an answer from the Association from Parent Products. What about cell therapy studies around the world as treatments for DMD? Well, we've now heard that this is quite a multifaceted issue, but what about the reaction of a family regarding this? So my first answer is, well, first of all, please come to see us and get ready to accompany all parent associations because they may truly help you understand what the general situation is like. We are all out there to find a treatment. But of course, having said that, maybe the experts want to provide their answers.
I'm not sure if I understood the question is about what is available currently.
Secondo me la domanda è come facciamo?
Well, actually, the question is how can families understand whether what is suggested they should do should actually be done? Sometimes their hope is to go abroad for a miraculous, treatment. So how can families understand whether this is right or not?
So maybe I can take this one. So first, it's important to go to meetings like this virtually or in person. There's also online information from parent projects like the Italian one, but also others. And then unfortunately, there are no miracle cures. So if you find someone who promises that they have the solution to Duchenne and often also many other diseases, it sounds too good to be true. It is too good to be true. So don't fall for this. Don't go to China or Russia or South America for a miracle stem cell treatment. It's a hoax. And even if you think, well, it cannot harm, I can try, it will harm your wallet a lot. And there's much better things you can do with that money and with that time. And hopefully there are no risks, but often you don't know what they will inject you with. So just don't do it. And if you have questions, you can always contact your patient organization, your parent organizations. They will be able to help you. And if they don't know the answer, they have a network that they can reach out, and then we will help find these answers. But don't fall for it. If it sounds too good to be true, unfortunately, it is too good to be true.
Yeah, I want to.
Grazie, Nemiche. Rita, sì, vuoi aggiungere qualcosa?
Yeah, I just want to support these comments. I mean, there is too much tourism on stem cell therapies, and you can find treatments anywhere. And as you saw from our conversation today, it takes a long time, and even 15, 20 years, you don't even know if you're going to develop. But, if there is something miracle, it's just not real. So just talk to the parent project, to your doctor. Do your research before adventure, and opening your wallet is not worth it.
Grazie Rita, anche a te. E rapidamente, poi magari un attimino.
Thank you, Rita. Okay, well, we may close with some final thanks, but in case you want to go to the parallel sessions that will be starting at 6:30, please do so, because the topics may be of some interest to you. So just visit the website and click on the live session that you're interested in. Otherwise, we'll meet again tomorrow at 4:30 because there's going to be another video, another beautiful video, and then we'll have a session on gene therapy. Having said that, let me take this chance to thank all our speakers, outstanding speakers. Thank you very much. Thanks to you all. I would also like to thank Filippo. By the way, we wouldn't be here without Filippo. I'll have to say that, And thank you all. I'd like to thank all those who are watching us from their home. We feel like you're close to us in spite of going virtual. Okay, Filippo, thank you very much. Thanks for giving me the chance to meet again so many people who've been on my side all my life. But anyway, it's not my fault. It's Pat Furlong's fault. She is the one who started it all. Thank you very much. Thank you.